My name is Mason Victors. I'm currently a founding fellow at Recursion and have previously served as the Chief Technology Officer and then the Chief Product Officer here for close to 4 years. If you are familiar with Recursion, you have likely heard mention of us navigating maps of biology and chemistry. We've been able to show demos of our maps and our operating system to a select few that have visited us at our headquarters, but now we want to show this to a larger audience. So, what does it mean to navigate our maps of biology and chemistry? In drug discovery, people try to navigate complex biology and chemistry to identify the most direct path from disease to healthy, but this is often limited by the relatively tiny fraction of biology that we actually understand or think we understand. There is no way to step out into the truly unknown or ill-understood areas of biology to explore them. At Recursion, we believe if we equip our scientists with a base map of biology and chemistry, a less biased and a more holistic survey of hundreds of thousands of biological and chemical perturbations in a human cellular system, that we can then enable them to explore the uncharted areas of biology to navigate the path to new medicines more effectively. Our maps shine light on the previously unknown and unexplored recesses of biology, and they serve as a tool to help us navigate just like regular maps, except these maps predict trillions of relationships across biology and chemistry to discover novel disease-relevant insights. We believe this approach is turning drug discovery from a trial and error process into a search problem, backed by what we call our virtuous cycles of atoms and bits, which is our ability to rapidly generate and analyze follow-up experimental data when our scientists find novel relationships amongst disease-relevant biology. Now, I'm going to walk you through an example of how we navigate rapidly from maps to insights to confirmed activity and finally to a potential drug discovery program. As a quick side note, for reasons of intellectual property and clarity, we'll walk through this process using an area of known biology. Our teams at Recursion are using these same methods and approaches I'm showing you today to delve into novel and unknown areas of biology to generate and prosecute new drug discovery programs. We'll start by examining the relationship of two disease-relevant genes with other biological entities. TSC1 and TSC2 are two genes that provide instructions for producing two proteins that work together to help control cell growth and division. Mutations in these genes can lead to a loss of protein function and can cause a biological system to go a bit haywire in a number of disease pathologies, such as tuberous sclerosis complex. If we enter these two genes into our map application query, we can discover relationships these two genes may have to each other, as well as to any number of other genes profiled by the Recursion operating system. What is revealed on the right is our version of a heat map, and it simply shows the inferred relationship between the queried genes and other biological entities. Each entity shown here is a CRISPR knockout of a gene as captured by Recursion's phenomics experimentation, turned into a mathematical representation with our proprietary deep learning algorithms. These mathematical representations of biological or chemical entities are compared to each other to assess similarity or differences and can be used to discover novel relationships that have yet to be documented in the public literature. When the heat map shows dark red, that shows a highly similar relationship between two entities. For example, this shows that knocking out the gene TSC2 causes a highly similar holistic cellular response to knocking out TSC1, as well as a number of other genes. We also see that knocking out either TSC1 or TSC2 shows a highly opposite holistic cellular response to that obtained when knocking out genes such as mTOR, Myos, and others. Those familiar with this area of biology will recognize that the RecursionOS has recovered many components of a signaling pathway here, without any prior knowledge of that signaling pathway. The only inputs here have been the images of cells where each one of these genes has been knocked out independently, and yet the map recovered these particular relationships in a very clean way. As these maps cover most of the whole genome, many of the genes that are returned when querying the map will be unfamiliar to our scientists, and so we provide a number of tools to enable them to quickly gain some understanding of the biological entities returned. While relationships between genes are rarely understood well, we can gain some quick information about what is known about each individual gene, its known functions, and diseases it is known to be associated with. Since we are returning not just individual genes, but rather collections, we can perform statistical analyses to identify pathway or protein complex enrichments, combining our proprietary map results with known biological data. In this case, we see that the area of biology returned by our query of TSC1 and TSC2 centers around TOR signaling. When setting up a query, one can imagine seeing mixtures of known expected relationships as well as novel unexpected relationships. That is when our scientists get really excited, because it helps them feel confident that our maps are simultaneously faithful to what is known or assumed to be canonical, while at the same time revealing novel relationships that have likely never been hypothesized, seen, or explored before. And that gives us an opportunity to dig deeper into those biological relationships to start to bring potential treatments forward on the journey towards patients. Having now explored some genetic relationships, we then turn our attention to relationships between these genetic perturbations and small molecules profiled in our systems. We can query to see which compounds show either oppositeness to the knockout of TSC1 or TSC2, or high phenotypic similarity to the knockout of mTOR. Examining the results, it is easy to see that the vast majority of compounds shown here are well-known mTOR inhibitors, some PI3K inhibitors as well. With this list of compounds in hand that show strong relationships to TSC1 or TSC2 knockout, or strong similarity to mTOR knockout, it is easy to quickly design a follow-up experiment to confirm these relationships. So, instead of screening our full library of hundreds of thousands of small molecules, we can instead focus on just the few dozen that show the strongest relationships to interrogate those further. This is done by designing and executing an experiment using our design software component of the RecursionOS. Since we have already done this, I'll simply pull up the previously executed confirmation screen. This provides a simple interface for our scientists to easily create, design, and manage the execution of additional experiments within Recursion's platforms, and is designed to handle the scale of experiments spanning over 1,000 1536 well plates per week, spanning millions of individual cell culture experiments at a time, with appropriate controls and plate randomization logic naturally built in. After running this experiment, it is easy to dive into the results and interrogate the data. Using one of our applications for analysis, we can dig into the results of a more traditional screen of compounds on the disease model, which is exactly what we have done here. After seeing the clear pheno-opposite and/or pheno-similar relationships between small molecules and our query genes of TSC1 or 2 and mTOR, we have conducted an experiment where these compounds have been tested directly on cells where TSC2 was knocked out via CRISPR to assess their ability to reverse the morphological effects of the disease and bring things back to a healthy state. And, as is customary practice here at Recursion, these compounds were tested alongside many dozens of negative control compounds to ensure that we have results that we can be confident in. In these plots, we represent the untreated, healthy control cells as these green glyphs to the left, and we represent the cell cultures where the disease model gene TSC2 was knocked out as these red glyphs to the right. Our compounds show up as glyphs connected via lines, each glyph representing a different concentration tested, so that we can see dose responsiveness where present. The x-axis represents a combination of the primary morphological differences between our disease and healthy control states, and the y-axis represents a combination of all other morphological differences that have no bearing on the disease model. This can be thought of loosely as an off-disease effect score, representing changes to biology that are unlikely to be disease relevant. We see that we have many compounds that cleanly rescue the disease state back toward the healthy representations before they start to cause additional biological changes at their higher concentrations. And these compounds that successfully reverse the disease state are indeed the compounds we had identified as pheno-opposite to TSC1 or 2 knockout, or phenosimilar to mTOR knockout. Taking some of our more potent compounds here, we can follow up with additional assays, such as Recursion's industrialized transcriptomics assay, TREC-seq, where we can demonstrate that our selected compounds also reverse a complex transcriptional fingerprint of TSC2 knockout relative to chosen negative controls. This concludes our demo, but to summarize, we have taken a few disease-relevant genes of interest, analyzed the known biology around those genes, selected small molecules of potential interest to assess more closely, designed and conducted follow-up phenomic screens to confirm those relationships, identified compounds that continue to potently act on the disease model, and then studied those in an orthogonal assay, confirming that these compounds do indeed modulate the disease in question in a compelling way. Thank you for joining me today. Hopefully with this exercise, you can see how navigating our maps of biology and chemistry gives us the ability to lead the next generation of drug discovery programs here at Recursion.